Okay, thank you, Ellen, for the very nice intro, and thanks everyone. It's really great to be here. This is a really great community, and I'm hoping we can forge lots of interesting collaborations here. And thanks also to Deborah and Stefan for really setting the scene for me here and doing a lot of the work and explaining some of the background for what I'm going to be talking about. So I thought I'd structure this talk around adverse outcome pathways. Splitting into three parts: exposure events, mechanisms, and then the adverse outcomes. But I'm going to do this in a slightly odd order. I'm going to start at the end and then loop back to the start. And the reason for that is I've actually done a little bit of work in the adverse outcome and phenotypes area, so I'm at least reasonably confident there. But from there, I'm going to kind of deteriorate into areas in which really I shouldn't be talking about, but I'm going to talk about it anyway. So, starting with adverse outcomes, phenotypes. So this is some work we've done a lot of work on in the context of the Monarch Initiative and various other projects. And one of the areas we're interested in is being able to search by phenotypes. Now, phenotypes are slightly, kind of like complex, fuzzy, inchoate kind of things. If you want to search a database for a gene of interest, you can maybe search by a standardized gene symbol, or maybe if you're searching, across species, you can search by its sequence, and we know how to search by sequences, and there's actually a kind of like a formal theory underpinning that based on evolution and base pair changes over time and so on. But if you want to search a database, maybe it's a database with categorizing genetic mutations, or maybe some kind of toxicogenomic database, and you want to search for a particular phenotype or disease or interest, like Hadzovic syndrome. Now that database may not actually have that precise string in it, or even a string that looks remotely like that, but it may have some kind of like description of maybe a mutant mouse that has a constellation of phenotypic features that recapitulate some of the features of that disease. But just searching with that and doing string-based searching will not get you any results. So one of the ways that we use to kind of like improve findability, to go back to the FAIR thing, is to use ontologies. So I've always had people evangelizing about ontologies. I'm going to do it some more. So ontologies give you standardized descriptors for annotating your data, and it makes it easier to find that way. But it goes beyond just standards for vocabulary. It provides you a shared model of the world. And you can use that to basically kind of agree, not just on the terms, but on how things work, how things connect together. And you can use this for reasoning and inference, and we saw some really nice examples of that as well. And when you connect these things together, you have something that you could call a knowledge graph. So I'm showing on the right here a knowledge graph that we've developed using a number of different ontologies that we've built for different projects. So in the middle is a disease ontology called Mondo, the Monarch Disease Ontology, and it's a classification of diseases. And so you've got maybe classifications of diseases like primary ciliary dyskinesia, which is a ciliopathy. And what we've done is we've connected this to other vocabularies, other ontologies. So ciliopathies can be described mechanistically by their connection to the cilium, and the cilium is a subcellular structure that is described in the gene ontology, where you find descriptions of subcellular structure, subcellular mechanisms, and so on, as well as parts of the cilium like the atractinium arm and so on. And we also have the human phenotype ontology, where we can link the phenotypes of the different subtypes of primary ciliary dyskinesia to their various phenotypic features like chronic rhinitis and so on. So this gives you not just only a nice way of describing these diseases and phenotypes, but a way of linking these things together that give you some kind of like model of the underlying biological reality there. So so we've heard about ontologies. We also heard there's a large number of them out there. There's a repository called Linked Open Vocabularies, which spans ontologies and vocabularies across multiple different domains. There's 650 in there in BioPortal, which is an excellent resource for ontologies in the life sciences, biomedical sciences. There's 716 ontologies in there, a total of 10 million terms. And as the question that came up earlier, there's a lot of overlap in these terms. So if you just search for cancer, that is found in 75 different ontologies. Now we have mappings between these, but the mappings are of variable quality, and it's generally, in my opinion, a kind of like a substandard way to go, when in fact, what you could do is you could actually, sociologically organize and say, okay, well, let's get together and build ontologies that like overlap as little as possible, and instead connect together in a modular way. So we created something called the Open Biological Ontologies Foundry, and or OBO for short. And the main goal here was to really kind of provide some kind of structure where people could build nicely integrated modular ontologies. So a lot of this is really just taking standard techniques from software engineering that we've learned over the years how to build software. We started building just giant, monolithic scripts that didn't reuse parts of other scripts and they're impossible to maintain. Gradually over time, we learned how to develop software in better ways using engineering principles. And part of this is really just trying to take this over into the ontology realm and providing the structure for doing that. So a lot of it is technical, but also socio-technical as well, providing a way for people to cooperate and just providing the tools and best practices for either kind of like forging new ontologies or working with existing ones. And we actually have a workshop coming up at this year's International Conference on Biomedical Ontologies in August. There's still time to register if you want to come to our workshop on building and using these ontologies. And we now have a small amount of funding to support the OBO project. And so if you go to obofoundry.org, you can find descriptions of all the ontologies, or I think almost all the ontologies I'm going to talk about today. And you can also find things like, if you want to make a request of one of these ontologies, they should all have a tracker. So we encourage the use of things using software engineering tools like GitHub. So if you want to request a term from an ontology, you don't have to try and figure out how to email the right person. You can just make the request on the tracker. And so it's all nicely kind of like public and transparent and well traced. And so one of the aims with OBO is to provide a framework for making ontologies in a modular fashion. So you can see this as kind of like Lego little building bricks. And the way I see it, kind of, and down at the bottom in the foundational layers, you have your ontologies for describing things like chemistry and genomes. So we've got CHEBI, the chemical entities ontology, so the sequence ontology. And this is where you will go to find your descriptors for various different kind of like small molecules, macromolecules, and so on. And then gradually you can build up on top of these, and you have things like cell biology described in the gene ontology, cell types in the cell ontology. The Go also describes biology at a kind of supercellular level as well. And then based on top of that, you have things like your phenotype ontologies and your disease ontologies, which can be used to describe things such as adverse events. And so an example of this kind of modular construction is terms from HPO, the human phenotype ontology, like palmoplantar, hyperkeratosis, which you can see as a kind of building block in its own right that you can use to describe your data, but that building block can also be kind of like broken down into its individual subcomponents. So this can actually be described in terms of like this is a phenotype that affects the hand, it affects keratinization, and it affects a particular skin layer, a particular cell type within the hand. So you can use languages like OWL to describe how these things are constructed together. And what you can actually do is describe different ontologies for different species using the same methods. And this gives you a way of inferring that these two terms, even though they are phenotypes in different species, have some kind of like connections to one another. And we've used this to implement fuzzy phenotype profile searching methods. And the basic idea here is if I'm interested in a particular human disease, like Hatzilevich syndrome, and you have a list of phenotypes for that disease that are described using terms from the human phenotype ontology, possibly with frequency information as well, you can do a search across different model organism databases, and you can find matching phenotypes. Now, you're not going to find exact matches because the more phylogenetically distant that you go, the more dissimilarities you see in the phenotype. So the algorithm is designed to make use of the structures in the ontology to give you a scored match based on how similar it is. So, renal hyperplasia may be similar to duplex kidney in the mouse. They both involve abnormal kidney morphology. And if you go to monarchinitiative.org, you can do perform these kinds of searches across species using your phenotype profiles of interest. And we've actually incorporated this into variant effect prediction pipelines. We have a tool called the Exomizer, and the idea here is that you may have someone with a rare monogenic disease, or hypothesized to be a monogenic disease. You don't know which variant it is underpinning this disease. Now, there are many standard variant filtering pipelines that make use of genomic information to try and infer what the causative variant or gene is here. But most of these only make use of the genomic information. Exomizer will make use of the phenotype descriptions as well, described using ontologies, and it will make use of the Monarch knowledge graph to actually boost the signals for certain variants that are in genes that are in perhaps a model organism that give rise to a similar disease or phenotype to the patient of interest. And we've actually, working with groups like Genomics England, we've kind of like integrated this into their clinical pipeline. So, this is actually, contributed to solving a number of cases that were unsolved before. So, this particular patient, Georgia, Exomizer predicted the KDM5B gene to be the highly likely to be causative in this individual patient syndrome. And this was based both on the genomic information, but also based on a protein interaction network and phenotypic similarity between the patient's features and what was found in model organism databases. Okay, so moving from there on to exposure events. When it comes to chemistry, we've got at least some kind of well-structured language for being able to describe exposures and so on. So if you want to describe a chemical exposure, there's various different kind of structural ways of describing your chemical, or using smiles and so on. There's ontologies like KEBI that give you nice identifiers and a hierarchy for being able to describe these. But of course, some exposures are a little bit harder to describe using some kind of structured chemical language. If you want to describe something like exposure to some kind of socioeconomic circumstance or living in a food desert or something like that, that's not something you can just distill directly to chemistry. So we started this new ontology called ECTO, Environmental Conditions and Treatments Ontology. And it takes its kind of core data model from EXO. So it really models things in terms of exposure events, stressors, receptors, and the stressors could be chemical, but they could also be macroscopic, kind of material entities like smoke, or they could be social as well. And you can also model the route of ingestion, inhalation, and so on. We developed this mainly to expand our disease annotation pipeline to incorporate environment as well. We're hoping to apply it to other use cases as well. We're also using it to integrate, there's a lot of exposure data being captured in model organism databases as well. We wanted to provide a multi-species way of linking, say, zebrafish exposures to human exposures. The ontology isn't yet on OBO, but you can, this is a screenshot from the Protege editor at the moment, so you can see it's got certain terms in it like dietary aflatoxin exposure. And these terms are all kind of built up in this kind of modular fashion. We're developing web-based tools to make it easier for people to request terms from this ontology. You can find these on the ECTO site. And we also developed some tools to help constructing ontologies in a modular fashion. And so this is kind of echoing what Deborah said. So in general, you never want to build your own ontology from scratch, but you may want to build a little domain-specific ontology that reuses parts of other ontologies. So we've developed something called the Ontology Starter Kit that makes it easier for you to create a GitHub repository and get set up with an ontology, importing parts of other ontologies. And so ECTO uses this framework, and really it's, in many ways, ECTO is quite a trivial combination of existing ontologies. So the real work has been done elsewhere, primarily in the KEBI, chemical entities ontology, but also in some other ontologies, such as we use Uberon, which is an ontology we developed originally for kind of bridging human gene expression data and model organism gene expression data, but we're now using just as a general standard reference for tissue types, organ types, and so on. And it's well connected with other OBO ontologies. So you have concepts in Uberon like the lung, which are connected, with structures like the swim bladder and zebrafish, but both by virtue of being respiration organs, also by virtue of being homologous to one another. Is connections to embryonic types like lung bud and so on. So you have the full ontogenic history of the different types as well, and connections into the gene ontology and other ontologies as well. So and this can be used for the describing the target organ for exposures. And ECTO also really builds heavily on another ontology that we developed called the environment ontology, or ENVO. And ENVO was really originally developed for describing metagenomic samples. So you take a sample and sequence it, and maybe it's a sample of water from near a kind of iron mine or something like that, or a soil sample, or a marine sample or something like that. But gradually over time, the use cases have expanded, and now there's descriptions of other things in ENVO as well. We've got complex materials in there like coal, oil, smoke, aerosols, all things useful for capturing exposures. Also environmental processes as well. So, degradation of soil and climate change and so on are modeled within that. And this is an example of how something like a volcanic eruption process is modeled in ENVO with connections to the various different components and the things that it emits. And I think we might have a volcano ontology expert in the audience. So this maybe looks quite naive, this representation. But so what we're aiming to do is connecting ECTO to disease and phenotypes. So currently within Mondo, we have connections from certain diseases like asbestosis to asbestos exposure and so on. But and these ones are fairly kind of trivial, but we're also collecting other annotations to other common diseases. So far with the human phenotype ontology, we've focused primarily on monogenic diseases that have very kind of like characteristic developmental phenotypes, but we're moving into the area of common diseases. And of course, there we want to model not just the genetic etiology, but also the environmental etiology as well. So we'll be releasing more annotations to ECTO in the future. And so finally, moving on to mechanisms. So this is a screenshot of like an adverse outcome pathway. And so when I originally saw some of these, my initial thought was, okay, this is a great area for being able to kind of use ontology to describe the different elements of these AOPs. And I plan to talk about that. But then recently I discovered that all this awesome work had been done in AOP Wiki. And although you can't really tell it just by looking at the wiki itself, but if you actually download the underlying XML, this is actually, many of these actually are using ontology terms under the hood. Now, not all of them, there's quite a few in AOP Wiki that don't yet have these ontology terms, but I was really pleased to see it's using Uberon and the Go to describe parts of these. But I think there's some parallels here to what we're doing within the gene ontology, which I'll talk about briefly. So historically, we saw yesterday how the Go is used in enrichment analyses. And so, most people in molecular biology are familiar with it. And one reason for the success of Go is it's had a very simple underlying data model where basically a gene is modeled as a bag of terms. So the BFA1 gene is just modeled as, okay, this is a gene that's got, it's in the spindle pole, it's a GTPase inhibitor, it's the bottom exit from mitosis. But the limitation of this model, it does not describe how genes work together to carry out some kind of biological objective. So we've devised a new data model that we're calling GoCam, for Go Causal Activity Model, where we can actually connect together the annotations in a causal graph. And so this describes the general structure, and this shows what new annotations for BFA1 and TAM1 would look like, where we can actually measure the causal flow. And this is, we have a very kind of activity-centric view of the world, where activities regulate other activities. So the GTPase inhibitor activity of BFA1 inhibits the GTPase activity of TM1. And so this is a much richer model. We've devised new ways of displaying these graphically. We've developed a tool called Noctua, that's a web-based collaborative tool. So lots of people can be working together on the same model. You can actually see who's editing the graph at the same time. The underlying representation is RDF-AL. We use a reasoner called Arachne, and a rule-based reasoner under the hood. that actually shows you in real time while you're working on a model, the actual inferences that you get from it. And we're working with integrating with Reactome pathway databases. It's designed primarily for describing the normal wild type function of genes, but you can also model, the effects of drugs like imatinib in it like this. And one thing we just started just very recently that I want to talk to Caroline about is modeling CTD events in the GoCam framework as well. So within CTD, you've got these very nice descriptions, describing how different chemicals interact together to give rise to, differential gene expression and so on. So we can actually model this directly. So we've got transformations going from the XML that underpins this into these models. We have a site that you can go to preview some of these. So it's not the normal Gene Ontology site. If you go to geneontology.cloud, you can browse some of these causal models. We don't have any of the AOP type ones in there yet because they're more experimental. But you can see our production models on that site. And so I think I'm about out of time. So yeah, just to summarize. So again, to echo what Deborah said, ontologies are absolutely necessary for fairness. But going beyond the fair as well, I would add a P in there for kind of like predictive modeling and reasoning, which they can be used for as well. And again, if you are building an ontology, please follow modular design patterns, reuse existing ontologies rather than building the entire thing from scratch. And this gives you a lot of benefits, not just reuse, but also wider integration. And we're using this pattern, for example, for exposure ontology. And AOPs can be represented quite naturally using some of the same design patterns that we're using in the gene ontology. So we should definitely work together on that. And there are way too many people to thank for all of this work. So I'm just going to put their names up there and leave it to questions.